Good morning, ladies and gentlemen. This is the joys of being a speaker first morning after the banquet. But I guess it's better than being the last speaker of the conference. I apologize for the very long title. I had to say something to remind myself what I'm talking about. It's been a very nice conference. My thanks to Jerry Pollack for enabling me to speak to you. And actually, my thanks also to Michael Seifra for making me realize there's this conference going on. I have a few heretical things to say to you. But one of the things I've heard over the last 2 days, people talking about pH and water, made me go back to something we did 25 years ago. And it was to do with mass transport and diffusion through liquids. And in this case, throughout my talk, all I want to tell you is that I think through Fick, first law and second law, flux of molecules through barriers, and voxel concentration changes through fluxes, I think we can use molecules as a structural probe for solvent. That's my hypothesis. So let me tell you how far I've got to. But having heard people the last few days, 2 days, I pulled out something we did, as I say, a long time ago. And I think I would say to you as a water community, you have a challenge and a difficulty. I cannot see how you can characterize water if you don't know its pH. And if you attempt to measure its pH, it's a lot more difficult than you think because you can't do it in deionized water. You have to do other things to it. And I tell you why pH is so important and a sensitive part of water. We were looking at mucus and water as barrier layers, as thin films. And this is what we showed 25 years ago. This is pH. And these are diffusion coefficients for hydrogen ion. So hydrogen ion is very pH sensitive in its mobility. For 25 years, I didn't know what was going on here. Nobody's shown this U-shaped curve. But I think there's a small possibility that right here, in the middle, there may be a different water organization. That's all I say, just a possibility. And if that's the case, if you're studying water and your pH shifts all over the place, and in pure water you have no buffering, trust me, then I don't know what you're doing in analyzing water. I actually belong to a totally different community, and it's the community of biosensors, and we make miniature biosensors and other biosensor devices which are kind of like that, which are membrane-coated, which contain enzymes, and all I'm interested in the metabolites, monitoring metabolites, and I do it in vivo. And there's some glucose going up and down in blood and tissue. That's all I do. But important to us in all of this is the membrane design, and I'll show you the relevance of that. The membrane design dictates the response of my system, in this case to jumps in glucose. And the dynamics of that tells me something about molecular transport through membranes. And I'm going to show you how we've exploited that to analyze motion through water. So this is one adapted design. It's a recess tip electrode. So there's gold here, 100 microns, and there's a recess. So I've trapped water, and I can look at mass transport of a molecule through water based on its electrochemical activity. And all of that's governed by Cottrell, except in our case, this is a stable volume, and therefore the Cottrell equation tells us our responses, bang, 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 for peroxide, for example, are stable. Now, what I'm interested in is the dynamic of that response, because the dynamic of that response tells me the one thing I'm seriously interested in, which is the diffusion coefficient of a microsolute through water. And that's, I hope, the relevant thing from your point of view. Now, Fick's laws may look very, very simple, but if you try to do exact computational analysis, it falls apart. You either need a very high computational power or the measurements are inaccurate. We've done a compromise by using two concentration in bulk concentration in a particular part of the water area in two different time zones, which then can be together to give us a joined up, modeled response of our electrode. So that's a model of the dynamic response of that little recess tip electrode. And one of these is the model, and the other is the actual electrode response. So when you have identity of the two, you can pull out, somewhere out of this system, the diffusion coefficient. That's all I'm interested in. So, for example, if I have a sensing surface more and more recessed inside that recessed tip electrode, and I make the diffusion distances longer, then, for example, for the lactate enzyme lactate sensor which has an enzyme in it, I can extend the analytical range of that finite enzyme reaction and pull it out and out and out, simply because I made diffusion distances longer. Generally, interestingly, the diffusion coefficients through that recess are pretty much as they are in bulk water, bar one type of molecule, anionic molecules. And the weird thing is that my device and its recess has got no real charge on the inside. So what is going on with retardation and ionic molecules? So this is a model system to try and take a look at. So we kind of studied that, but I wanted to then adapt it to something that has simply shocked me after 30, 40 years of research. And what shocked me is that we had a project to look at one important membrane barrier, which is the blood-brain barrier. It's an absolute resistance to nasty drugs and toxins to protect your brain. And all the biologists say is to do with the cells around the blood-brain barrier. Our hypothesis was that the basement membrane at that barrier may just be important because it's a kind of membrane in its own right. Generally, the interest of membranes is because they're artificial, we make them for hemodialysis or whatever, or tissue matrix and so forth. And I'll give you an example of that. All we were trying to do was to create a tissue-engineered blood-brain barrier. And to do that, we used a very standard methodology. This is a culture, two-compartment culture system. And there is a porous polymeric membrane. And the one that everybody uses is track-etched membranes. Now, the beauty of track-etched is the holes in it are big, linear, non-tortuous, and you can grow cells on them on one side as well as the other. And bingo, we have a blood-brain barrier. Or that's what I thought. So these are the membranes that we worked with, and it's the sole reason I'm here talking to you, because of the membrane science. They are beautifully, elegantly organized in terms of a quasi-Gaussian distribution. But the important thing is that with the right imaging technique, you know absolutely the overall pore area of the membrane. If you know the pore area of the membrane, then you know what to expect in terms of diffusion coefficients, because it's like bulk water diffusion, except through a finite pore area. And these are micron-sized pores. So you expect bulk equivalent transport. Right. This is where we ran into a serious problem. Our experimental setup is there. There's the membrane there, and it's protected by dialysis membranes, and we do electrochemistry stuff on it, just like that dynamic modeled response that I showed you. And we picked the easiest and simplest small molecules. These should not be affected by anything on that pore diameter size. Pyrocatechol, cytomyophen, ascorbic acid, and peroxide. It couldn't get easier in terms of electrochemistry. This is baby electrochemistry. It cannot go wrong. Or can it? Yes. Yes. Well, okay. My experiments are better at, let me tell you. So we have this system, and we did what I told you about based on the Cottrell equation. You look at dynamic response in MOLVAB. But we also picked up the standard approach for kids' school, kindergarten electrochemistry, which is cyclic voltammetry, where it uses zoom voltages back and forth, and using a particular equation form. And the only thing I was interested in is the peak current here. And that peak current gives you diffusion coefficients if you know everything else. So we have two methods that look at diffusion coefficients through membranes trapped along here. That's the only deal in town. And let me show you what we found. This is bulk water diffusion coefficient. They're same order of magnitude, but they do vary based upon the size of the molecule. I get that, and that agrees with the literature. There's nothing special here. If we take the dialysis membrane, I'm sure a lot of you experienced those, what you find is, this is the first thing ever done by anybody on this, is that you do get diffusion, which is pretty open. This is for water. Ratio between water and through the dialysis should be 1:1. Actually, it's a little bit out, and it does go down. But the model response, is very, very precise and reliable. So we have reasonable diffusion coefficient. Okay, everybody happy so far? Fine. And I can work with my blood-brain barrier with these membranes. And then we got into a shock, and I criticized the people who did the work for many, many months. Now I realized they were doing the right thing. Let me show you what we found. These are diffusion coefficient values, and they are for the polyethylene terephthalate membrane, which was one of the track etch membranes, and polycarbonate membrane. And if you look at the diffusion coefficient values here for peroxide, this material has zoomed down diffusion coefficient. This is totally not acceptable, totally counterintuitive. It's also got diffusion coefficient values for the other molecules, which are much, much lower. But these are micron-sized pores. This is not credible, not possible. And there's also resolution based on large and small pore size, which is just weird. But let me give you the punchline to this. The punchline is that if you take the ratio of the diffusion coefficient through bulk water to diffusion inside these dirty, great big pores, what do you see? You see pore ratios of what? Less than 10%, 0.025. I mean, this is like 2%, less than 2% of what you see in bulk water. This is not permitted, and that's what we saw. Not only that, this material acted as less of a barrier than polycarbonate, and yet both polymers are uncharged, nominally inert, and that's why biologists use them. So this is a serious, serious shock to us, okay? All these molecules are going through these pores, and water, it says, I've had enough, I'm going to block this stuff. And I don't understand why, after so many years, what is going on. All right, so let me just give you that punchline so that you don't forget numbers. The numbers here should all be 1. Bulk water diffusion equals water diffusion through these open channel pores. And look at the numbers. They are out of this planet, and they're different, especially with ascorbic acid. There's anionic rejection, and there's size-based rejection, and it's rejection period anyway. How much of this is artifact? Well, this is, where am I going? Yeah, this is the jump technique, the amperometry that I referred to, and this is cyclic voltammetry. They don't entirely agree. They're close enough to be experimentally valid. So that's the first thing. The second thing is that you're still back to zooming down of diffusion coefficients based upon which molecules you're dealing with. This is serious, serious selectivity and resolution for membranes that everybody uses. Biologists have been using, since the First Testament, and nobody's bothered to look at diffusion through them. It's fantastic. Now, so where's the artifact from? Potentially you could have blockages inside the track edge material. People who've studied this have found actually the pores are barrel-shaped. So in fact, we've underestimated porosity by looking at the surface characteristics, surface features. So this is seriously has to be looked at. And I'm just emphasizing the fact that this material, the pore area sequence goes like that, but this material then jumps the queue because diffusivities are materials-based. So why is that happening? And I don't have an answer. But we got it under the radar, and it's published in a pretty good paper. So there's our material, and that's what I was born with and what I was grown up with. Yes, the double layer. The double layer stops within, 0.01 microns of a surface. Yes, charge fields. And water, of course, is immobilized and semi-loose and bulk water, and you got slipping planes and the kind of Helmholtz layer and all of that. there should be nothing going on way out there. So it cannot be the physicist's double layer. I'm really tempted to say to you that I think water structure in the micropores is different to that in bulk water, and that's what we're going to investigate, having found these results. And I think the materials have a long-distance effect, which is not dissimilar to, I guess, easy water, except this is not so easy. And I think there are implications right across biology, because everything inside the cell is in close proximity to a surface. And the reason why we picked up and had precision analysis of diffusion here is because this is one of the only membranes which has no tortuosity. Everybody that works on membranes has to work with tortuosity. And they make a guess as to how tortuous the pores are. These are straight, linear pores, and there is no hiding place. Okay, I'm going to frame shift and give you some results on biology which are more flaky, but I just want to share them with you. And I'm not sure how much is water and how much is biology materials. Let me tell you what we're trying to do. I'm back to the blood-brain barrier, which I think has a polymer barrier, which is connective tissue. So extracellular matrix. And so we decided to work on collagen 1, collagen 4. Collagen 4 is the basement membrane collagen. I'll tell you about it in a minute. And we were interested in adding to it various things which are already present in your connective tissues, heparin and chondroitin sulfate. Interesting because they're negatively charged. And we want to look at concentration effects of the collagen itself, ask the question, is it charge selective? And actually, you know and I know, proteins are stereoisomers, so were they enantioselective? This is cartilage, totally avascular. And none of us in cell biology really know why the cells inside are not dead. They're a long way from a blood supply. And possibly why in your tendon your cells aren't dead, because they have a very limited blood supply. So let me try and show you what we found. This is what collagen 1 is meant to do. It's fibrous, it's structural, it's a scaffold. This is collagen 4. It simply acts as a carpet for cells to sit on everywhere. And when we immobilize, I'll show you how we do it, immobilize, you get a different kind of fibrous versus non-fibrous structures. They are structurally different. Now, here's one form of cheating that I've done. There's no other way around it. I can't isolate these membranes and these polymers just like that. So what we do is we take a bifunctional reagent, glutaraldehyde. Some of you may have worked with it. It's a wonderful agent for cross-linking protein, creating amide bonds, and bingo, you've got a nice membrane. So we take this membrane and used it as a barrier, just as we did with the track-etched membranes. And here's the kind of stuff we found. So sort of as expected, with collagen 1, for example, as you raise concentration of the protein barrier, you start to get reduced diffusion coefficient. Nothing special about that. I no longer know what the tortuosity is in there, so I don't know how much is water affecting things, how much is tortuosity through these membranes. But I'll show you a couple of things that make me sit up. This is kind of interesting. That's collagen 1, that's collagen 4. 1, 4, 1, 4, 1, 4. So the same amount of collagen protein creates a different barrier effect. That tells me something about the importance of 4 as a barrier structure. I think there's biology to be done here, so I just want to share that with you. What I cannot tell you is what is and what is not water. But we came to yet another difficult, anomalous answer. Here's how it goes. These are ratios of diffusion through the materials, the collagens, and this is diffusion through bulk water, and the diffusion ratios, well, you don't expect that to be 1 anymore because you've got a barrier, fine. But so you're about, 20% of what you should be. That's not the point. The point is that for each microsolute, the ratios against bulk water are different. Now, that's really not viable because these protein barriers we've created are not microsieves, they're not molecular resolution things. And yet a microsolute, the hydrodynamic radius of this is probably 50% of that. That's nothing. And yet you've got a massive difference. So is there something going on here? I don't know. Well, I think there is, but this is rather more understandable, thank God. It's a test for how valid our results are. So this is collagen heparin, and collagen chondroitin sulfate. It's approximating to the kind of tissue matrix that we have in our body. For a neutral molecule, there's not a lot in it when you throw in anionic species. But for ascorbate, which is anionic, you can zoom down its diffusion using an ionic polymer inside the collagen. So that tells me something, that our measurements at least are valid, and on this occasion, reasonable. Okay. This is heparin, this is chondroitin sulfate, and they're charred species. That's fine. That's all I need to tell you. And you're back to the depression of ascorbate diffusion through heparin and chondroitin sulfate polymer. And this time, these are ratios. Again, as I've shown you, zoom down permeability, you zoom it down, but here it's starting to get charge-based selective, and here this ratio and this ratio and this ratio, I go back to my point, should not be different. These are microsolubles, and these materials should not have any kind of effect. So there's one kind of molecular sieving which may or may not be due to solvation water around these proteins. But there's one other magic trick that biology has up its sleeve. So in biology, in my neck of the woods, in tissue engineering, we attempt to recreate biological tissues. And mostly we fail because we can't make tissue which is thicker than about 1 millimeter. Beyond that, inside, the cells are dead. There's no circulation. So how does cartilage, 3, 4, 5 millimeters, have living cells inside? I don't know how it does it. And the gurus, I think, don't know how it does it. But here's an interesting diffusion coefficient outcome. This is natural cartilage right here. And these are diffusion coefficients around the 10 to the minus 6 centimeters to the minus second for all these species. They're kind of like dialysis range in spectrum, 10 to the minus 6 order. And this is dialysis membrane. You can read bulk water. They're also 10 to the minus 6. So despite the having a whole load of collagen junk on board, diffusion through cartilage, natural cartilage, is pretty damn high. And I ain't figured out why, and nobody has yet looked at that. So that's a takeaway message for what is water doing or not doing. This is the other interesting outcome, which is that we took tendon, the stuff that I'm walking around with. Now, it's got collagen, which is about double that of cartilage collagen. But the diffusion coefficients are now 10 to the minus 8. They are 2 orders of magnitude lower. It defies some serious understanding. What is more understandable is that orientation has an effect. So my tendon is linearly orientated. So if I measure diffusion that way versus that way, I have different tortuosity. And probably that explains why, whether I'm parallel, anti-parallel, I am getting different diffusion coefficient values. That's understandable. But it's, what is the barrier? Is it the protein? Is it hydration shell water? I think the jury is still out, and I think it's worth studying. What was, in a way, sort of interesting is that we took, only one experiment, we took tyrosine in D and L form, okay? Mirror image, enantiomer, going through collagen, which is itself a stereoisomer. So what do we see? Actually nothing. D and L diffuse in the same way. Is that clever? Well, maybe, maybe not. What it might tell us, what it just might tell us, is that any diffusion retardation doesn't involve selective binding with the collagen as the molecule moves through. It may be just because it's having to traverse water. So those are the findings that we made up. This is pretty much the end of my story, but let me tell you this. I think we used 2 methods which gave us reliable diffusion coefficient. We have a unique multi-point fit for diffusion coefficient determinations. I don't think the data is junk. What I can't tell you is what's causing it. And it has to break some taboo. The taboo is what everybody in my business says. The surface water is different to bulk water. Yeah, we get that. But in my field, we also believe there is loose-bound gel water. Tanaka's one of the people who said that. Gel water creates a kind of stabilized interface for proteins and cells so they don't rupture and denature. And a lot of people in my business have used polyethylene oxide, very hydrated, end-attached polymer, and it creates a kind of quasi-aqueous environment, which we know makes for very biocompatible surfaces for proteins. They are great probes for what water is doing right there. So that's the story of yesterday. What we've observed, I think is valid, is telling us stuff is going on way, way out there. But I remind you that the key thing is that you can't make these studies unless you control pH and you know what the pH stability is. Otherwise, the whole water experiment, in my view, is a waste of time. So, porosity doesn't predict permeability. Biology must do it in a very clever way that we don't understand. We have a problem with the double layer because it's only the start of the story, it's not the end of the story. And we have serious problems of trying to understand what cell biology is doing intracellularly, because I think the water there is seriously solid. And that's pretty much what I wanted to communicate to you. Thanks for your attention.